To study a proteome, the first step is to disrupt the cell or tissue so that the protein molecules can flow around freely in solution. At this point, it's important to emphasize that when one refers to a protein, we're not talking about one molecule of a protein, but hundreds or thousands of identical molecules of a protein isoform. This realization has important consequences for proteome analysis because the more molecules of something that you have, the more likely it is to be detected and produce high quality data that you can have confidence in. All of the molecules of the same protein isoform are chemically identical, and thus they all stay together during fractionation and analysis. The majority of organisms that exist are mainly made up of water, and all biological molecules have to interact with water molecules. The 20 different amino acids that make up proteins interact with water in different ways. Some have ionic interactions like the acidic and basic amino acids. Some form hydrogen bonds, while others are hydrophobic and would rather associate with other hydrophobic molecules within the protein and without than interact with water. Because proteins are long chains of amino acids, the chain can be folded up so that the hydrophobic regions are no longer being exposed to the water molecules. Hydrophobic regions of a protein can also be associated with hydrophobic regions of other proteins, forming protein complexes, or with lipid membranes, and thus excluding water molecules in both cases. Unless the protein molecules can freely move about in a liquid, the protein cannot be separated from other proteins, a necessity to reduce the complexity of a sample so that as many proteins as possible are identified and quantified. However, many proteins are poorly soluble, meaning that they don't stay in the solution or liquid, and can precipitate out of the liquid, meaning that they can't be analysed. Proteins that are embedded in cellular membranes are one such example, and if they can be removed from the membrane, the hydrophobic or water-hating regions in each molecule can associate together with each other and with similar regions from other protein molecules, whether they be a molecule of the same protein or whether they be a molecule of a completely different protein. The mass of molecules associating becomes larger and larger until it precipitates and falls out of solution. If we simply took a cell and broke it open in a physiological buffer solution, such as phosphate buffered saline or PBS, and centrifuged that sample to bring all of the large insoluble components to the bottom of the tube, the liquid or supernatant would contain a large number of proteins that are normally found in the cytosol of a cell, and these proteins can be easily fractionated by chromatography or electrophoresis to reduce complexity. However, the pellet at the bottom of the tube also contains a large number of other proteins which can't be further fractionated, and thus our analysis of the proteome is incomplete. So how do we access this part of the proteome for analysis? The trick is to use other chemical additives that stop the protein molecules from associating and keep them in solution, but they don't interfere with their fractionation or analysis, such as this detergent molecule. Thankfully, there are a great number of additives that can be used, but great care and planning must be used because some of these additives are not compatible with some fractionation techniques, while others can interfere with downstream analysis. It needs to be noted that proteins often exist as complexes, where numerous molecules of the same or different proteins come together to execute their function. These complexes can fall apart or dissociate if certain chemical additives are present, such as these surfactants. Again, careful planning is necessary. Often a chemical additive is added and then needs to be removed again at a later step to prevent it interfering with a certain technique. The chemical additives can be divided into a few groups. The first group we'll look at are Coatropes. These are small molecules that are able to disrupt hydrogen bonding with water and other amino acids to unfold the protein chain to expose hydrophobic regions that can interact with the Coatrope molecules. Thus, the Coatropes can also break apart protein complexes. Coatropes are usually found present in the highest concentration possible without the Coatrope precipitating, up to 8 molar for urea. If the Coatrope needs to be removed, it's most often achieved using either dilution, dialysis, or precipitation of the proteins. But it must be noticed that upon Coatrope removal, it is very unlikely that all the proteins will still be soluble in the non-Coatrope solution, and some will be lost through precipitation. The next group of additives that we are going to look at are the surfactants, or surface active agents, and these are similar to the detergents that you would use in dish or clothes washing. These molecules have one end that can be ionized to interact with water, and another part that's hydrophobic that can interact with the hydrophobic regions of proteins. When surfactants bind to a protein molecule, they can often disrupt or open up and denature their structure, unfolding the regions of the protein and disrupting protein complexes. The most widely used surfactant is sodium dodecyl sulfate, or SDS, which has a strongly anionic group and can solubilize most proteins, including membrane proteins. However, its strength is also its weakness, and its strongly anionic nature means that it cannot be used for separating proteins by charge, something we'll return to. To overcome this, a number of surfactants have been produced that are zwitterionic, having both an anionic and cationic site in the molecule that allows interaction with water molecules, but means that their overall charge is zero, and thus they won't move in an electric field like SDS. There are also a number of mild surfactants that won't unfold proteins or disrupt complexes, but instead bind to the surface exposed regions of the proteins, stabilizing them when they're taken out of their native environment. Surfactants are very difficult to remove from proteins, but there are some methods, mainly using ion exchange chromatography or organic solvents. What should start to be apparent is that these additives are acting like intermediaries between the proteins and the liquids that surround them and they float in, preventing the protein molecules or complexes from associating together and aggregating into large masses that can fall out of solution, such as this example where the surfactant's hydrophobic tail is interacting with the proteins and their polar head group is interacting with the surrounding liquid. But solubilizing proteins is just one part of the problem, the other part being breaking the cells open in the first place so that the proteins can be accessed and then solubilized. With samples of mammalian cells that are surrounded by lipid bilayer membranes, breaking them open is relatively simple, and often just adding Coatropes and surfactants is sufficient. Cells with rigid cell walls, such as bacteria, yeast and plants, are far more difficult. The best way to break these cells open is to use either mechanical means to smash them apart, or use ultra-high pressure or ultrasonic sound waves to burst them, or enzymes that specifically target the cell wall. One of the most effective ways is to use a ball or bead mill with a sample that has either been snap frozen in liquid nitrogen or lyophilized to remove the water. The dry sample is placed in a liquid nitrogen cooled metal canister with a metal ball, sealed and shaken back and forth really fast. The metal ball smashes the cells into the sides of the canister and shatters them. This is performed until the sample has a consistency of fine talcum powder, which can then be suspended in liquid with different mixtures of buffers, Coatropes or surfactants so that the protein molecules will go into solution. So in short, if you can't break the cell open efficiently, you can't access all of the proteins, and if you can't get the proteins into solution to be analysed, you will end up with an underestimation of the proteome.